It's my pleasure to invite Dr. D. Satyanarayana, presently working as Associate Professor of
Dr. Ashutosh, we are not able to hear you.
Ashutosh, audio on, please.
It's on. Is it on now?
Yeah.
Yeah. Sorry, the audio is not on. It's a pleasure to invite Dr. D. Satyanarayana, presently working as Associate Professor of Microbiology at Rangaraya Medical College, Kakinada. He graduated from Osmania Medical College, Hyderabad. Later did his post-graduation from Gandhi Medical College, Hyderabad. Later did his fellowship in medical virology from National Institute of Virology, Pune, and completed his Harvard Medical School T2T program of medical education. He worked on various positions at Nizam Institute of Medical Sciences, NIMS, later SWIMS, later Osmania Medical College, then Colonel Medical College. He is a convener of Medical Education Technology Unit of RMC and principal investigator of ICMR Virology Laboratory at RMC Kakinada. He has various publications and chapters, and I invite him to the session. Sir, you have 20 minutes time.
Yes.
Request you to stay on time because we are running much behind the schedule.
Sure, sir. Thank you very much, sir. First of all, I would like to thank the organizers for giving me this opportunity to present this session in the AP Neurocon 2020 in the session of Dr. S. Balaparameswara Symposium 2020. In the next 20 minutes, I will be talking on the virological aspects and molecular diagnosis of SARS coronavirus 2. I will be covering the virology, overview of the pathogenesis, and also some of the aspects of the laboratory diagnosis of SARS coronavirus 2. As you all know, this coronavirus disease, COVID-19, the organism, the pathogen causing it is named as severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, according to the International Committee on the Taxonomy of the Viruses. This belongs to the family of the coronaviruses. This family is not new to us. Many of the coronaviruses are infecting us for several decades, but they are causing mild infections so far. But the ones among the genus beta coronavirus are causing a potentially life-threatening infections. Among them are the SARS virus, which we had previously, and Middle East respiratory syndrome related coronavirus, that is MERS-CoV, and now we are having the SARS-CoV-2. All these belong to the genus beta coronavirus in the family coronaviruses. As the name indicates, all the coronaviruses will have a corona-like appearance. That is, a spike-like glycoprotein gives a corona-like appearance, and it also has M protein in its membrane in the envelope, and hemagglutinin esterases, and RNA and N proteins, and E proteins. These will comprise the structure of this virus. And of this, the envelope of this virus is derived from the host cell membranes, over which the spike glycoproteins synthesized by the virus are projecting out. It is a positive sense single-stranded RNA. That means the RNA itself can be translated directly into the different proteins. And it is a non-segmented genome, and it has about 30 kilobytes size. And because it is RNA virus, the mutations do occur because of errors in the RNA replication. RNA always have a error-prone replication strategies. That is the reason why the new variant strain is emerging. That is because of the mutations in the S gene. The new variant is that is recently identified is being named as variant under investigation. That is VUI 2020-12/01. Now, a paradox is actually it is easy to kill this virus, and it is difficult to kill this virus. Because it is an enveloped virus, it is almost susceptible to almost all commonly used disinfectants if you properly use them. But at the same time, it is also highly resistant to environment. It can stay on surfaces, especially smooth surfaces, for even days. So the transmission is primarily because of the respiratory droplets, and if the risk is more, if the activity is less than 2 meters, and also formats. For example, so door knobs and also the tap handles, all those in the communal areas, especially in the communal areas, because it is surviving in the smooth surfaces. Aerosol transmission is also noted, and the transmission in the other routes is under investigation. Now, this SARS-CoV-2, this S protein, that is a spike protein, binds to the AS2, that is angiotensin converting enzyme 2 on the target cells. And this AS2 is present in several diverse areas on the cells. For example, most of the type 2 pneumocytes will have this, and also conjunctiva. That is the reason why rubbing in the eyes even can transmit this infection. And intestinal epithelial cells, that is responsible for GIT symptoms, nasal goblet cells, olfactory epithelial cells, renal epithelium, endothelia, and also in the CNS. Now we'll see the basic aspects of the SARS-CoV-2 replication, because it helps us to understand the different targets for the drugs. Once the spike protein identifies the AS2 receptors on the human cells, it binds to it, and the membrane of the virus fuses with our host cell membrane, and this virus nucleic acid enters into the cells, and the RNA genome is released. The RNA genome translates into different proteins, and also it gets transcribed. It gets packed in our Golgi complex and endoplasmic reticulum, and the mature variants will be released. So these are the different areas where we are already having the drug targets. For example, the monoclonal antibodies, or the convalescent plasma, or the human recombinant soluble ACE2, all these when administered, they will block the virus entry into the cells because they attach to the AC2. And proteolysis after translation of the that happens once inside the cells, that is blocked by leponavir and ritonavir. And the replication itself, that happens because of the RNA dependent RNA polymerase enzyme in the viruses, that is blocked by that is the famous drug, that is remdesivir. And so many drugs, the non-specific drugs we are using, for example the chloroquine or hydroxychloroquine, that we are using, that interfere with the TLR activation, that is non-specific activation of our immune system, as well as this part assembly part also will be interfered by those drugs. So these several application steps helps as the targets for drugs. And once we see the pathogenesis, once the S protein binds to S2, virus enters the cells and replicates. While it is replicating, the virus releases so many of its proteins into the cell. The extra proteins, that is not human proteins in the cell, will be presented by MSC class 1 molecules to our immune system. At the same time, virus also, what it will do is, some of the viral proteins don't regulate the ACE2 on our cells. As a compensatory mechanism, once the ACE2 is down regulated, there is overproduction of ACE2 that increases the vascular permeability, that causes hypotension, that leads to lung injury. In parallel, because of the viral proteins getting presented to our immune cells, there is activation of alveolar macrophages and release of pro-inflammatory cytokines. For example, interleukin 1, interleukin 6, interleukin 12, TNF alpha. Because of the aberrant release of these pro-inflammatory cytokines, eventually will lead to the cytokine storm. This cytokine storm is like a vicious cycle. There will be a dysregulation of the human immune response with increasing viral load. As the virus infects one cell, millions of copies of the virus will be manufactured in that cell. And that cell again releases all those millions of copies of the viruses, which will infect all the adjacent cells. With the increased replicative cycles of the virus, there will be further dysregulation of the human immune response. And there is excess release of the pro-inflammatory cytokines. That itself will lead to the recruitment of neutrophils, macrophages, monocytes with activation of the T cells. And there is inflammation and tissue injury with reduction of the surfactant. That leads to the ARDS, that is a major manifestation, pulmonary manifestation. And there is pulmonary, vascular, and endothelial cell death, diffuse thickening of the alveolar wall. And because the pro-inflammatory side, diffuse thickening of the alveolar wall along with the endotheliosis, that results in the ground glass opacities in the CT scan. And because of the cytokine storm, all these cytokines also will be released into the circulation. Once they are released into the circulation, they will try to damage the other organs. And also there is activation of the coagulation cascade as an immunopathogenic mechanism that leads to shock as well as multi-organ dysfunction syndromes. This is a brief pictorial depiction of the immunopathogenesis. And one important thing here to note is in the initial stages of viral replication, when the virus infects our cells afresh, for some time it suppresses type 1 interferon gamma response during the initial infection. You know that interferons are actually interfering with the viral replication. Virus have some proteins that can suppress the interferon gamma response so that it can happily infect the neighboring adjoining cells. That is one thing. And also because of the delay in the interferon response, there is a delay in the onset of symptoms. That means the patient will continue to replicate, the virus continues to replicate in the patient's body even before the symptoms do appear. By the time the symptoms appear, most of the cells in our body are already infected by this virus. Now, that neurological manifestations of the SARS-CoV-2, I will briefly summarize. Actually, it was discussed in detail by the previous speakers. The infection of AS2 positive cells happens in the nasal or olfactory mucosa. There will be delay in the external transport of the signals that leads to the loss of smell and taste. There is a cytokine storm that injures the blood-brain barrier, and with the injury to the blood-brain barrier, there is entry of cytokines into the brain that leads to seizures and encephalopathy and headache and other things. And there is several neurological manifestations also because the ACU2 receptors also present in the several parts of this central nervous system. And there are certain issues in the immunopathogenesis which are still controversial and we need further understanding. For example, importance of the immune response in the severity as well as recovery. Will it help or will it aggravate? We don't know yet because it is a complex phenomenon. For example, females have the robust immune response when compared to males, but males will, and also that means we expect that severity is less in females. In the same way, if it is because of cytokine storm, the older people will not have much of immune response, but severity is more in the older people. So there are several controversies regarding the immunopathogenesis, which we have to understand further. And also there is a molecular mimicry. The virus, some of the molecules of the virus will mimic our own cells that leads to immunological damage. And another controversial thing is if there is immunological memory from the previous coronavirus exposures, because as I mentioned, we are already exposed to the several types of other coronaviruses previously, which caused mild respiratory or GI tract infections before SARS itself. So will they cross-react and will they enhance or will they protect us? That also has to be understood. And what is the role of neutralizing antibodies? How long they survive? There are several studies state that some will survive only, antibodies stay only for a few weeks. Some will say that antibodies will stay longer. And also, what is the role of genetic constitution? This also has to be further understood because same family, some people are getting severe infection, some got infected and recovering well. And is it because of any comorbid conditions or another genetic constitution also plays a role along with the comorbid conditions? That is also being investigated. So this is a timeline of investigation. As you can see in this picture, in the pre-symptomatic phase itself, we'll have so much of viral genome, and all these stages already discussed by previous speaker. So I'll skip this part. Now, once we understood the composition of the virus structure, what parts will help in diagnosis? For example, our targets in the diagnosis as well as therapy will be like the nucleic acid proteins, membrane glycoprotein, spike glycoprotein, envelope glycoprotein, and the replicase proteins, that is ORFs, open reading frame 1A and 1B, these will help in the replication. And one more thing that is present inside the virus is that is RDRP enzyme, that is RNA dependent RNA polymerase enzyme. Many of these serve as targets for viral diagnosis. So the samples we do collect for diagnosis will be preferably nasopharyngeal swabs and oropharyngeal swabs, the Dacron or polyester flaked swabs that will be transported in the viral transport medium. The other specimens that may be tried are the bronchoalveolar lavage fluid, tracheal aspirates, nasopharyngeal aspirates, nasal wash, and sputum tissue, that is biopsy or from autopsy from lungs, that are transported in the VTM again, and serum, that is paired sera, acute and convalescent phases sera, whole blood, stool samples, urine samples, all these can give the diagnosis, but we preferentially do from the nasopharyngeal swabs and oropharyngeal swabs. And as I mentioned earlier, normal swabs that we use for microbiology diagnosis in the bacteriology are not suitable for viruses. We have to specifically use the Dacron or polyester flocked swabs. And they should be transported in the 4 degrees centigrade with the triple packing, and they're stored at 4 degrees centigrade if they're processed immediately. If there is delay, we store them at minus 70 degrees centigrade. Now, our diagnosis depends on 3 important areas. One is the RNA, that is detected by molecular tests. Suppose are the viral proteins that will be detected by antigen assays, or our immune response to the viral antigens, that is in the form of antibodies, that antibody detection. We'll see briefly what are the available tests. Among the molecular tests, the gold standard is the real-time RT-PCR test, and there are some modifications for this RT-PCR test, that is cartridge-based assays. That is the famous TUNAT test, or even the gene expert that we routinely use for TB diagnosis. That machine can be used for RT-PCR for conducting COVID diagnosis also. And there are some newer methods. I will briefly tell what are they. And antigen detection, antibody detection, and virus culture. That is only for research purposes. We are not routinely doing it. Coming to the real-time RT-PCR, that is RT-PCR means reverse transcriptase polymerase chain reaction that happens in the real time. The sample that is collected in the viral transport medium that reaches the laboratory from where we extract the viral RNA. That is the step called RNA extraction, and that RNA will be converted to the cDNA, complementary DNA. That step is called reverse transcription. Because the polymerase chain reaction takes place, we need a DNA as a starting unit. So we have to convert the RNA to the cDNA. That happens by reverse transcription, and that proceeds to the polymerase chain reaction. In the old time, after the polymerase chain reaction is over, we have to detect the amplicons or the amplified products by running the gel documentation. But over the last 1 decade or so, the real-time PCR became famous. That means as and when the amplicons are available, with each cycle, we can detect them by the fluorescence. That's why it is called as real-time PCR. So total it is real-time RT-PCR, to say it correctly. All these procedures will be conducted in the BSL-2 laboratory. In the real-time PCR, once the viral RNA gets amplified, after certain cycles, for each cycle it doubles. 2 becomes 4, 4 becomes 8, 8 becomes 16, like that. After certain cycles, it reaches the threshold of detection. That is called CT value or cycle threshold value. So for screening, we use only one gene, that is usually envelope gene. For confirmatory testing, we are using 2 or more genes, that is ORF gene or RDRP gene or ESRN. All these genes can be for confirmation. But nowadays, we are not doing the 2-step procedure. We are doing the multiplex assays. Simultaneously, we are detecting either 2 or 3 genes at single time. This is the importance of the CT value, where we can detect the threshold when the crosses the detectable limits of the fluorescence. Now, there are certain challenges and the limitations of this detection. One is the quality of the specimen and transporting and handling issues. It should be transported in the cold chain. Timing of specimen collection. Very early, for example, today you are exposed to a patient. Immediately, if you give the sample, it will become definitely negative, false negative. So it is very early in the infection or late in the course of infection. Both times you will get the false reactions. The false negatives do come. False positives do occur. It is rare but very possible. In the laboratory, we take all measures to control the false positives by in-house controls. There are certain mutations do happen in the virus genome. If the major mutations do come, we may fail to detect by the available RT-PCR. We have to have the advanced kits. So TrueNet is actually a cartridge-based testing, the same principle, but everything happens in the small cartridge. Okay, that is TrueNet. It is also a singleplex or multiplex. Nowadays we are doing only multiplex only. The newer techniques that are available are the RT-LAMP, reverse transcriptase loop mediated isothermal amplification. That is a recently available technique. And RPA, recombinase polymerase amplification, that is one more technique. And the CRISPR based techniques are became famous nowadays, and they got approval by FDA and also in India also. And nanoparticle technology by rapid visual detection without requiring any advanced machines. By visual color change itself, we can detect by nanoparticle technology. In Hyderabad also, the biotechnology units are developing the kits using the nanoparticle technology. And next gen sequencing. the sequencing of the viral genome. That is now what we are doing now, because of the some cases are coming from UK which are turning positive. They are to be sequenced. For that, we are depending on the sequencing machines as well. And nucleic acid mass spectroscopy, that is actually a very costly affair and is not available at several places. So I'll discuss only about one thing that is became famous nowadays. That is FELUDA. That is based on the CRISPR technology. CRISPR means clustered, regularly interspaced, short palindromic repeats. The Tata Medical and Diagnostic Limited developed this test. That is Tata MD Check CRISPR test. That is FELUDA means FON CA9 editor linked uniform detection. All these are dependent upon the genome editing techniques. These are developed by Council of Scientific and Industrial Research and Institute of Genomics and Integrative Biology and brought to us by Tata Medical Care. They are approved by DCGI for use in India. And FELUDA is one of the detective in the Bengali literature. So it is named after him actually. That acronym correlates with that. And they have 96% sensitivity and 98% specificity when compared to the RT-PCR. Coming to the rapid antigen tests, nasopharyngeal swabs are used. It is a point of care test. Results will be available within 15 minutes, but sensitivity is less despite the high specificity. And rapid antibody tests are not dependable always because the kits are not validated properly, and they can be only of utility after a minimum of 7 days after the onset of symptoms. So for acute diagnosis, we cannot use them. And this is a timeline of different things where the PCR is becoming positive and antibodies when they become positive. These slides are taken from the JAMA. And all results are uploaded onto the ICMR portal and state government portals. We regularly update them as and when the results are available. Each day we are processing about 3,000 to 4,000 samples in our laboratory. So all these protocols are available on the ICMR website. Those interested in depth study of these protocols can download them and study them from the ICMR website itself. So the strategy is also changing from time to time. These are also available in the ICMR website. And there are about 4 groups: routine surveillance in the containment zones, routine surveillance in the non-containment areas in hospital settings, and testing on demand. For the first category, what is surveillance in the containment zones, the first test preferred is antigen test, but it can also be followed by RT-PCR test. But the other 3 categories, as and when available, RT-PCR should always be done in preference to the other tests. So to summarize about the diagnosis, RT-PCR is sensitive, gold standard for diagnosis. Rapid antigen test is specific but not very sensitive. Antibody testing is useful only surveillance and epidemiological purposes. And patients with high index of suspicion who are negative by rapid antigen test should be retested by RT-PCR. And also remember that if the patient with high index of suspicion who is negative by RT-PCR also, maybe a test may be repeated after a gap of 2 or 3 days to say whether really he is positive or negative or positive. So to rule out false reactions. And in the laboratories, we always follow the ICMR strategies for testing. So with this, I think I conclude my presentation. And thank you all for your patient listening. And once again, I will thank the Neurocon for giving me this wonderful opportunity. Thank you.
Thank you, sir, for a wonderful lucid lecture. Few questions invited.
Yes, sir.
Can I ask you one question?
Yes, sir.
Murthy,
Sir.
this RT-PCR, what is the sensitivity percentage, and rapid antigen, what is the sensitivity percentage? Like sometimes you get false positives and false negatives. Then how to go about it? Like,
yes, sir. Actually, rapid antigen is highly specific, sir. It is very rare to get false positives in validated antigen test kits. The specificity is more with antigen test kits, but sensitivity is very less. We will miss the actually true positives also. In case of RT-PCR, the sensitivity is very good, sir, about 96 to 97%, and specificity is also equally good, 95%, if you follow the stringent protocols. In the laboratory, we always run the positive controls, internal controls, and also the negative controls. Actually, there are mechanisms to increase the specificity also. Normally, the specificity ranges from 90 to 96%. So for RT-PCR. Yes, sir.
Can I ask a question?
Yes, sir.
Dr.
Sure, sir.
So Dr. Murthy, excellent lecture.
Sir.
So my question is something, which every physician or every surgeon is thinking.
Correct.
We are coming up with the possibility of a second spike.
Sir.
And all of us are thinking that will we be able to treat elective cases, or still we have to continue the same lifestyle and all that.
Correct, sir.
What are your suggestions to create COVID and COVID-free spaces? So let us say it's possible for us to create a COVID-free space where the surgeons or physicians can handle patients without any fear, or they can do surgeries without any fear. Because when I spoke to some of my colleagues in China, they are doing transnasal endoscopy. They said, "Oh, we don't bother. We are just wearing masks and the usual OT stuff because."
Sir. Sir, actually, there is a, like for HIV, we have a dictum. Every person is, we have to treat him positive unless proved otherwise. In the same way, in the COVID also, unless proved otherwise, every person, when you are outside, you should treat every other person is positive. We should always, there is difficult to have a non-COVID space in the present scenario. So we have to follow all precautions whenever you are doing all the invasive procedures. I think you covered very well in the usage of PPS in the general areas as well as in the intensive care areas. Actually, that protocols, if everybody adheres to those protocols, I think we'll be safe. Otherwise, it is very difficult to predict the COVID scenarios nowadays because many are asymptomatic. That is a paradox we are seeing nowadays.
Now, the other question is, many of our patients have got infected by healthcare workers.
Okay.
Now, most of the patients who are admitted, they are undergoing RT-PCR within 72 hours of admission, and then if required, we are doing a second time. And we have had patients after surgery, they became COVID positive because they got most probably from some healthcare workers. So, are you thinking of recommending any kind of prophylactic screening for healthcare workers?
Yes, sir, definitely, sir. All healthcare workers, frontline warriors should be actually screened for COVID. That is one thing. The second thing is all should be given training properly, sir, for hand hygiene and also following the protocols.
I know, but nowadays we are not, at least in AIMS, we are not screening all the healthcare workers.
Yes, sir.
I was just asking you, is there any ICMR policy, or are you aware of any other?
Yes, sir, there are ICMR policies, the strategies. Healthcare workers have to be screened. They can be screened. Actually, we are screening all our doctors, all our staff, but they are not routinely done or regularly done because we have the workload in the regular hospitals. So that is one thing. They can be screened, sir. Actually, healthcare workers can be screened periodically. I think every 14 days they can be screened. But irrespective of the screening, better to adhere all the prophylactic protocols, sir. The screening helps you to have the RT-PCR results, but that will not help you to stop the spread the infection because false positives make false negatives to come more than the false positives. So rather than depending completely on the screening tests, it is always adhere to the protocols of hand hygiene and other things.
Thank you.
So this is Dr. Varaprasad. One thing is for emergency surgeries, we are using RAT, rapid antigen test, RAT for emergency surgeries, and elective RT-PCR. So what you suggest for emergency cases and elective cases? What are the tests?
Sir, that question is to me, sir.
To you only.
Actually, for all the emergency cases, in emergency, you can depend on the RT rapid antigen test, sir. If it is positive, it is positive. But the problem is the sensitivity is less. Yes. In parallel, you can also send a sample to RT-PCR and request the laboratory specifically to process it on the priority. Normally, in our laboratory, we have a protocol, sir. All the surgery cases and any antenatal cases waiting for delivery, the samples are processed in emergency in the next available run. Normally the turnaround time will be from 16 to 24 hours, but all the emergency samples at EIT will be about 8 hours only. So we can follow the rapid antigen test, but negative test does not rule out the infection. You can always have the backup of RT-PCR. So there is nothing wrong in collecting 2 swabs at time and in parallel sending for both.
So in India, we are not able to take up what Sarath Chandra told. It's not possible to take all those precautions. So India Institute, okay.
Correct, sir.
Go to periphery, all these precautions we are not able to take it up.
So proper hand hygiene and mask, these two will help us to reduce the burden of 90% to about 90%, sir.
Especially for inpatients and
yes,
surgeries,
sir.
what I say,
yes, sir.
Inpatients and surgeries.
These two are minimal things for the healthcare workers, sir. Proper hand hygiene and a mask.
So RAT followed by RT-PCR, same protocols you are going to advise.
Correct, sir.
Simultaneously, we have to take and test.
Yes, sir.
Can I answer to what? Can I make a comment if you don't mind, sir?
Sure, sir. Please.
So that's an excellent question asked. That
Yes, sir.
how can we follow these protocols in all the other hospitals? So difficult.
Correct, sir.
So I would advise that
Sir.
the simplest is obviously you have to do for emergencies RAT. So you have a separate COVID suspect place or a COVID suspect ICU, where unless the RT-PCR proves negative, you can keep the patient there. So that is mandatory. That has to be done.
Yes, sir.
Second is a donning and doffing areas have to be created. The second thing is also very, very mandatory.
Correct, sir.
Third is RT-PCR for all electives at least 72 hours before the surgery.
Yes, sir.
Which is mandatory.
That is a, 1 minute. There is actually a protocol of ICMR also, sir. The patients admitted for elective surgeries, ICMR itself designed the protocol. They should have tested by RT-PCR.
Now, once this is done, the OT, once the intubation is over, wait for 20 minutes. And
Yes.
once the draping is over, you're more or less safe when the
Correct.
surgical area is exposed.
Yes.
Then you can take off your goggles and you can operate regularly under the microscope.
Yes.
And if somebody doesn't have the PPEs, I think the best alternative is remove your OT clothes in the OT, have a good bath, wear a fresh set of clothes, and then go home.
Yes.
That is
Sir. Correct. Exactly, sir.
In the present scenario,